the branch chain amino acids. The initial step in the degradation of branch chain amino acids is a transamination reaction. In the second step, the alpha keto analogues of these amino acids undergo oxidative decarboxylation by alpha keto acid dehydrogenase complex. The alpha keto analogues are converted to their CoA derivatives, which undergo dehydrogenation. As a result, beta methyl protonyl CoA is formed from leucine, which leads to the formation of beta hydroxy beta methyl glutaryl CoA, which forms acetoacetate and acetyl CoA. Methaacryl CoA is formed from valine, which undergoes series of reactions to form succinyl CoA, and tiglyl CoA is formed from isoleucine. It finally converts into acetyl CoA and propionyl CoA. So that's the formation of methacrylyl CoA, and here are light green rings at the back of the slide to signify the introduction of the double bonds in these compounds through dehydrogenation. The degradative pathway for valine and isoleucine has two major functions: first, to generate energy, and second, to provide precursors to replenish the TCA cycle intermediates. Valine and isoleucine contain carbons that form succinyl CoA. So here we can see that leucine is exclusively ketogenic with formation of acetoacetyl CoA and acetyl CoA. Valine is glucogenic through formation of succinyl CoA, and isoleucine is both glucogenic and ketogenic, and it forms acetyl CoA and propionyl CoA. Propionyl CoA can be converted to succinyl CoA. During these degradative reactions, NADH and FADH2 are generated for energy production. One NADH is produced during the oxidative decarboxylation of each of the branch chain amino acids. Besides, one FADH2 is produced when isovalyl CoA is converted to acetoacetate during degradation of leucine. One FADH2 and two NADH are produced when isobutyryl CoA is converted to propionyl CoA during degradation of valine, and one FADH2 is produced when isovalyl CoA is converted to HMG CoA during degradation of leucine. Now coming to the disorders. In maple syrup urine disease, the branch chain alpha keto acid dehydrogenase complex that oxidatively decarboxylates the branch chain amino acids is defective. This leads to accumulation of their keto acids, leading to intellectual disability, and there is burnt sugar smell in the urine. So, maple syrup urine disease may be classified as type 1A, which arises from mutations in the E1 alpha gene, type 1B, which arises from the mutations in the E1 beta gene. In both cases, alpha keto acid decarboxylase is defective. Type 2 is due to the mutations in E2 gene. The component affected is dihydrolipoyl transacetylase. Type 3 is due to mutations in E3 gene. The component of the carboxylase dehydrogenase affected is dihydrolipoamide dehydrogenase. Alpha keto acid is decarboxylated to form a hydroxyethyl derivative bound to the reactive carbon of the thymipyrophosphate. So that's the alpha keto acid dehydrogenase complex, and its first subunit is the E1, and it contains the TPP. Here the TPP is the coenzyme of the alpha keto acid decarboxylase. Next, the hydroxyethyl intermediate is oxidized by transfer of the disulfide form of the lipoic acid, which is covalently bound to the dihydrolipoyl transacetylase domain of the alpha ketoacid dehydrogenase complex. The acetyl group bound as a thioester to the side chain of the lipoic acid is transferred to the CoA. The sulfhydryl form of the lipoic acid is oxidized by dihydrolipoyl dehydrogenase, which is E3, regenerating the oxidized form of the lipoic acid. So to summarize, you can see that this is the alpha keto acid dehydrogenase complex. It has three components, E1, E2 and E3. In E1 there is thymine pyrophosphate. It binds to the decarboxylated form of the amino acid. Then the thymine pyrophosphate detaches and the amino acid combines dihydrolipoyl. In the presence of dihydrolipoyl transacetylase, it combines with the lipoic acid. Lipoic acid contains the SCoA in its structure, and this it transfers the SCoA to the keto acid formed through decarboxylation, and so the CoA derivative is generated, and the form of the in this process the lipoic acid becomes reduced, so its oxidized form is regenerated through E3 complex, which contains FADH2 and NADH, and the enzyme is dihydrolipoyl dehydrogenase. Now coming to the disorders. The disorder is the maple syrup urine disease. The defect lies in the alpha keto acid dehydrogenase complex. There are elevated plasma and urinary levels of the alpha keto acids, branch chain amino acids, and the alpha hydroxy acids. Signs and symptoms include fatal ketoacidosis, neurological derangements, intellectual disability, mental retardation, and maple syrup odor of the urine. Pyruvate after decarboxylation may combine with butyrate to form isoleucine through successive reactions, or it may combine with pyruvate to yield valine or isoleucine through subsequent reactions. The first three reactions of the degradation of branch chain amino acids are transamination, oxidative decarboxylation, and dehydrogenation. Leucine is exclusively ketogenic and forms acetoacetate and acetyl CoA. Valine is exclusively glucogenic and forms succinyl CoA, whereas isoleucine yields succinyl CoA and acetyl CoA. Branch chain amino acids serve as metabolic fuel as their catabolism yields NADH and FADH2.